Dr. Kazer, before we get into HIV, single cell genomics, or even Sanofi, when you were a college student, what did you think your life in science was going to be like?
Dr. Kazer is my father. He's a physician, so I'll just go with Sam for today.
Sure.
Um, I thought I was going to work for L'Oreal when I was a senior in college. I majored in chemistry, and this whole graduate school thing, that was on the table seemed really overwhelming. Um, and so I went to job fairs, and L'Oreal at that time was hiring, associate level chemists. And so I talked to them. I talked to a paper company, people who made manufactured papers for McDonald's. It's a lot of chemistry that goes into food wrapping, actually.
Yeah.
Um, and I also had potentially another opportunity to go to Switzerland, although they didn't want to bring me in to work on plastics there either. So I honestly didn't know, uh, science in my future at that point was completely a blank space, open field, and I knew I liked the idea of science and trying to figure things out, but I had no idea what I wanted to do with my life or how I wanted to engage in that in the long run.
I think a lot of people think that they have to have it all figured out by a certain point, and ultimately, throughout all these conversations, I'm finding more and more that pretty much no one has it figured out, and we're all just kind of winging our way through life and hoping for the best.
Yeah, I mean, I think there's so many guardrails that get put on or, suggested to you as a high schooler, as a college student, as a graduate student. And, the intentions I think are always good in terms of trying to guide young professionals into a career path that will be successful and rewarding for them because there are so many steps that you have to accomplish to go forward in various career paths, especially in science and technology, right? Um, but at the end of the day, there are many, many avenues forward. And the hard part is understanding what those are. I think that's the big challenge right now for young scientists, especially in this current era, post COVID. How do you know what's even out there? What can you do? Because I think your scope is so limited in terms of what you're exposed to in right now in academia.
And it's got to be tough for those people because I feel like it changes every day what's possible and what's not possible. And if this kind of honest conversation is helpful to you, hit the subscribe button. It helps the channel and it lets me know to keep making more episodes just like this. Obviously did not go to work at L'Oreal. You did not work at a paper company. In fact, after college, you graduated from Columbia, you went to MIT to pursue your PhD. And that was a pretty big pivot, even from just what you've described. And then I know you were also pretty into physical chemistry at the time. What you ended up changing into was remarkably different. You ended up going into working on single cell genomics, global health, HIV. What clicked for you during that time that made you think this is the scale that I want to think at and this is the work I want to do?
If I'm going to be totally honest, I didn't know that that's what I wanted to do, and things sort of shook out. And I've become a big fan throughout, especially in my most recent transition, which I think we'll talk about a little bit later, of just like following the track and seeing where the road forks and understanding, when it's time to make a change. When I went to MIT, I had very strong feelings that I wanted to work on materials chemistry, as you can tell based on like what I was doing. And as you said before, I used to be a physical chemist. I used to shoot lasers at things and measure dynamics. I met Alex Shalik, who is a faculty member at MIT, now the director of the Institute of Medical Engineering and Sciences, who was a physical chemist who was turned biologist, basically. And I had the option when choosing my graduate lab to basically continue in a physical chemistry lab or join Alex Shalik's lab and sort of take a hard right turn at Albuquerque, like Bugs Bunny might say, and, try something different. And I think because I was so unsure about whether or not graduate school was what I wanted to do in the first place, it seemed like sort of taking this step into the unknown was something that could be fun, exciting, and different. And it quickly became clear to me that global health was something that reinvigorated my passion for science in a completely different way than what chemistry and materials chemistry had done initially.
Yeah, so you mentioned global health. And specifically, I know some of your work had to do with HIV, and especially HIV at basically the earliest possible moment before the immune system even know what's happening to the body. What surprised you most when you finally saw those data at single cell resolution?
Yeah, I think HIV is a really interesting disease because it's a model infection that we have a lot of insight into, because it is been so prioritized, um, by the U.S. science, uh, machine, if you call the NIH that. But um, I think what really surprised me about looking at HIV at the earliest time points was, one, how different it is for every person. Despite being an infection, right? We know how the virus looks and what that looks like, how completely different this response and how it looks in different people. Um, and then also how systemic and sort of broad changing, the response is, right? And it's an infection of the blood, as well as the immune system is part of your blood, and it also exists in all of your tissues, right? But it's something that can be uniquely studied in the blood, and so that has given us sort of like a really interesting opportunity to try and understand how early responses in individuals, right, those first immune responses in individuals, may dictate or inform or reflect how they're going to respond long term, and what their response to treatment might look like, or whether or not they're going to naturally control HIV infection, which a very small percentage of people are actually able to do without the need for antiretroviral therapy.
I don't think many people know, maybe I don't even know, even though we were friends in graduate school and even my roommate was in your lab. I don't think many people know what single cell RNA-seq is. Can you tell me what that is?
Yeah, so RNA sequencing is the idea of you're trying to read the RNA, from a tissue or a group of cells. And, in the central dogma, the biology central dogma, which I learned, on the job, right, is that DNA in the cell encodes RNA, which are messengers that tell the cell how to make proteins, and proteins are how cells do things, right? And so RNA is very easy to read because it has 4 bases, just like DNA. And so you can use sequencing technologies, which vary. Now most common sequencing technologies use fluorophores or lights, basically to read out which base you have from the RNA sequence. And you can read that out in mass, in parallel. And so you can read millions of sequences in a very short period of time. And so the idea with single cell RNA sequencing is that we're going to read all of the RNA from single cells independently. This was a big advancement, in the early 2010s, because of the increased capacity for sensitivity. The amount of RNA in a single cell is very hard to detect by old methods. And so in sort of molecular biology speak, there's been a big revolution in our capacity to amplify and measure. I make copies of and measure very, very small amounts of material. And so the upshot really is that what single cell RNA sequencing allows us to do is understand how individual cells are processing sort of a response to information and what they're doing. And then the broader picture is, can we use data on lots, thousands, millions of single cells to understand the ensemble dynamics? How does a tissue or an organism respond to some perturbation, some challenge?
Why HIV specifically? Like, to my knowledge, I think most people, probably at least in the United States, feel like HIV is a done deal. Like, we've already exhausted all of our efforts, and largely for many people, it is no longer a death sentence that it was back in the 80s, for example. So, can you tell me what was why HIV?
Yeah, I think, personally, I ended up working on HIV because I met some very cool people in South Africa who were trying to tackle this issue. And so for me, generally, science has been about working with good people. And the people who were working on HIV that my lab was connected with were good people. And so that was a draw. In terms of the, you know, HIV and the global health issue, is that it's still a persistent global health issue, despite first world countries like the US and many in Europe having HIV under control. As you said, it's sort of like a done deal in the US. So if you take antiretroviral therapy, which is a daily oral drug regimen, for life, you can have the same life expectancy as someone who has not been infected with HIV. You don't develop AIDS, right? You can't transmit it either, right? So you completely, um, free. However, if you stop taking those drugs, it will come back, right? It sticks around. But in non-first world countries and developing world, there's reticence to take these drugs. They're not necessarily easily and quickly available, especially now that the U.S. has retracted many of its international support systems, especially around HIV/AIDS. Um, um, and it spreads very quickly through largely through intercourse, right? But also through blood transfusions and other means.
Yeah. And since your work in that field, you've since moved into mucosal, respiratory immunity, COVID, nasal immune memory. Do you see a common thread between HIV and respiratory viruses that people miss?
I think what is interesting is that the immune response that someone comes up with to a virus is conserved in the broad sense, right? Um, Akiko Iwasaki and Ruslan Medzatov, who are two sort of like pillars in the immunology field, have done a very good job at sort of codifying the set of responses that the body has to infection, either viral, bacterial, helminth, parasite, right? And so these steps are often conserved. But the way in which that manifests and the way that which that plays out, if it's an infection of the blood, if it's an infection of the lung or the nose, or of the liver, for example, with hepatitis, right? Those steps play out in a different way. So the overall structure of that response is conserved, but the way in which it plays out and the way in which we have to treat it, right, or prevent it from happening, is slightly different, right? And I maybe we'll talk a little bit more about this, but where you get protection from is critical.
What do you mean by where?
Physically in the body, where are you developing protection, right? And so, in general, you create a vaccine so that you can develop antibodies that circulate and can respond to some infection. And so for something like HIV, it would be great if we had a vaccine because it's a disease of the blood. And so if you get a vaccine intramuscularly or subdermally, that's what most vaccines you get are, then that creates, circulating, i.e. it goes through the blood over and over again, these antibodies that can be protective. The problem with HIV specifically is that it mutates too quickly. It's just a very fast mutating virus. And so you create a vaccine, you get antibodies, and those can't catch up.
Yeah, what's the alternative to a vaccine that specifically creates an immune response?
In HIV, there's a couple approaches that they've been working on. So one, right, is there are these people who create broadly neutralizing antibodies. These are people who have been infected with HIV for 25, 30 years, and they naturally start to create antibodies because they've seen so many different iterations of the HIV virus that they're able to create an antibody that actually can block HIV entry into cells on their own. And so there are labs that are trying to basically, through serial immunization, lead or trick the immune system, the B cells, into creating antibodies that are broadly neutralizing without having to go through 25 years of exposure. The idea would be is you could get 3 or 4 shots, and we rationally design what's in those shots to basically trick the immune system into creating that broadly neutralizing antibody early. So that's the vaccine route, right? The other route is the cure route, which I think is a much harder problem because HIV can go dormant, which means that the reason why if you take antiretroviral therapy, that eliminates all actively replicating HIV in your body, but HIV integrates itself into the human genome. And so you have cells that have HIV integrated into their DNA, and they don't create HIV except sporadically, randomly, right? And so they mostly remain dormant. If they pop up, they get killed immediately during antiretroviral therapy treatment. But if you don't have that treatment constantly, those few cells that still have HIV in them can then re-replicate. And so a cure is very challenging because those cells that have HIV incorporated into them don't have any other identifying features that say, oh, we should kill those cells. They're not expressing anything on the surface, right? Um, and so genomic medicine, like really editing the genome stuff, that's challenging. And I don't know if you heard about sort of like the scientist in China who, created babies with CRISPR knockouts, but he disappeared, right? But that experiment, what he did with those babies was eliminate the co-receptor for HIV. It was the intention of basically saying we want to remove the capacity for HIV to infect somebody. So he, in those CRISPR babies, as they're called, removed
I
the
didn't even
gene
know
for
that.
that.
I
Yeah.
don't know why, but I thought that they were making just like designer babies, and that was the big uproar.
No, I mean, the initial experiment for that scientist who went missing was he knocked out CCR5, which is a co-receptor for HIV, with the intention of like, well, if your cells can't express that, then you can't contract HIV infection.
Does
But
anyone know what happened to the babies? This
I don't
was,
know,
I
actually.
think, 10
Yeah.
years ago at least,
Yeah.
something
Yeah.
like that. So,
I,
wow.
I don't know. I don't know.
Obviously, there's an ethical dilemma there, and largely we, as a scientific community, have decided we're not going to do that yet because we don't know the implications. But I'm still fascinated to know about this rogue vigilante and what ended up happening. I think most of the scientific community would be interested.
Yeah.
So, so you graduated from MIT and then went and did a postdoc. Can you tell me a little bit about that transition?
Yeah. So at the end of graduate school, I had the idea that I still was very interested in understanding infection and viral infection dynamics. As a former physical chemist, I was very interested in kinetics and dynamics. That's what you sort of learn about in those classes and in college, and specifically interested in how early stage dynamics and responses impact late stage um, sort of outcomes. Right. So do early responses impact late stage outcomes? And studying the blood HIV was very interesting and provided an opportunity for me to connect with very cool scientists in South Africa, learn a lot from immunologists and understand. But there's very little you can do to test things in humans, ethically and rightfully so. Now it's very challenging to do that. If there's a treatment available for people, you give them the treatment, right? There are
Right.
no more real control groups in severe infection or cancer cases, right? Um, and so I was interested in moving into something that had broader reach. And so in my postdoc, I wanted to work on nasal immunity um, because it's the entrance and the beginning, at least we think, for respiratory viral infections, which are the major, leading infection globally, right, in terms of where infections are happening.
And what year did you start your postdoc?
I started my postdoc. Good question. I started my postdoc in 2020. Um, and,
Seems like a good time to start studying respiratory infections and how they enter the body.
correct. Yeah. That, as much as I can say I didn't, intend to start my postdoc and study COVID, it definitely was at forefront of mind as I was making that transition. I actually chose to study the flu when I got started, specifically because it's something we could model in animals and something we had a lot of control over. Um, and I think what we found over time is that there's a lot of similarities in the initial infection period for a virus that has never been seen before between flu in mice and SARS-CoV-2 in humans.
And what did you learn during that postdoctoral experience?
Scientifically, we learned how the immune response unfolds in a tissue. And I can go into the gory details of it, but we sort of describe this stepwise response, codify kind of how the epithelium, the cells that line the nasal mucosa, get broken down, how they then attract immune cells to the tissue that create inflammation, that then attract adaptive immune cells that are able to actually kill the virus, right? And then create a long-term memory response. Um, in terms of like more broadly, what I learned during my postdoc is that, um, you know, there are core tenets of immunology and biology, and the interesting and fun parts of being a scientist are drawing those connections and
Mm.
understanding kind of like how does something I've learned in this situation or something that was described 100 years ago, like resurface and reconnect to the thing I'm doing now, right? And we talk a lot about trying to not reinvent the wheel in science, because there's a lot of focus, as you know, right? There's a lot of focus on doing new things
Right.
um, in science, right?
Right. So this postdoctoral experience was at Harvard Med School, is that right?
It was at Harvard Medical School in Boston Children's Hospital. So I set up my own co-advised postdoctoral experience with the intention of trying to leverage two things. So one, leverage mentorship and exposure that came with working for someone who was very famous. And so if you're an academic trying to become a professor, working for someone really famous turns out to be really important. Um, Unfortunately, the names associated with your mentors end up making a big difference when you're on the job market, because people like to see trainees who have trained with people they respect,
Sure.
and that way they know that they're going to be good scientists. But
Yeah,
then I
so your to put it succinctly, your career trajectory was Columbia for undergrad, MIT for graduate school, Harvard Med School, Boston Children's, postdoc, ticking all the boxes of a future academic. And now you made a transition into industry. Can you tell me about why you made that transition into industry, how you're feeling about that transition into industry?
I made that transition for a couple reasons. The first is that it's a poor time to try to become a research professor in America. Um, Science overall is being defunded and deprioritized, and be setting yourself up for a very challenging path. Um, the other part is that I'm a new father, and when you have a child that you have to take care of, the realities of your life completely alter. And I want to spend time with my family. And as a young academic, it's very challenging to make time for anything other than your career. And so making this move into industry, into pharma, big pharma as they call it, provides me with that opportunity while also giving me room for growth. And so I've really been enjoying this transition so far in terms of learning a different way to do science, but still has the same amount of, impact and interest as I did as an academic.
I think that's a really great story because I think so many people often assume that you make a decision, you follow your dream, you achieve your dream, and you stay on that path undeterred. And ultimately, and I try to have this conversation with my students all the time, is that they're so worried always about what they're going to do with the rest of their lives that they don't even stop to consider that many of the decisions that they make will be unmade by circumstances out of their control. As you mentioned, starting a family changes your perspective about things, whether or not you can devote the time and energy and resources necessary to pursue one career path versus a career that might be more stable and provide more for a family financially and also provide the time necessary to be an active participant in a family is a thing that, as an many of my students are 21 years old, they're not thinking about those things, right? um it's always fascinating to hear that ultimately almost everyone's life story is filled with these forks in the road that change their path. But ultimately, we all, hopefully find a way towards happiness in a way that maybe looks different than what we had initially intended. So I appreciate you sharing that story with us. So you mentioned this move to big pharma, as you put it, and as it's lovingly known in certain circles, and now you're at Sanofi. So talk to me a little bit about what Sanofi does. I don't understand for anyone who doesn't know and maybe listening. Typically, for people who work at these types of companies, you can't tell me exactly what you're working on. It's probably proprietary. Totally fine. Tell me, what does Sanofi do? What are they known for?
Yeah. So Sanofi chases the miracles of science. That's our current tagline. And right now, Sanofi is a very old company, a French company, and it's now a global company. Historically, we've been very involved in vaccines. And so actually, like a lot of people get Sanofi's flu vaccine every year, at least in the
Mm.
US and Europe. But in the recent, sort of in the last 5 years, we've become very well known for a drug called Dupixent, which maybe you've seen commercials for. And this is a biologic, meaning it's an antibody, monoclonal antibody that's used in an autoimmune situation, disease, right? And so it was initially approved for skin diseases, atopic dermatitis, but now is also being used for things like nasal polyps, as well as other autoimmune disease categories. And the way this drug works is it blocks signaling receptors for a certain type of immune response, the IL-4, IL-13, which are interleukins, which I won't go too deep into this. It's called the type 2 immune response. And so what we do now in the research and development department is try to identify novel targets and create drugs for these novel targets. Mostly right now, our portfolio is mostly centered around immune diseases.
Fascinating. So let me ask you this. I know every time someone on my podcast talks about working in science, the comments that I receive most commonly are ones that are people saying that pharma is terrible, pharma is bad, pharma is nefarious, and
Mm.
ultimately they just want to keep people sick so that they will keep buying their drugs and keep filling the pockets of the big pharma CEOs. What has your experience been like?
So what I'll share is my opinions are my own, and what I'm talking about today are things that I think and don't reflect what Sanofi's mission or statements are of it. What I have to say is that um, at Sanofi, our vision and our goal, and that's communicated to me, right, as an employee, is that we act for patients, and that what we do is to try and improve people's lives. At the end of the day, it's very hard to make a drug. It's very, very hard to make a drug, and it's very, very expensive to make a drug.
Why
And
is it so
creating,
expensive?
yeah, that's a great question. Um, so right now, on average, it takes about 10 years from the inception of a drug idea to get that drug, approved and into patients and prescribed. And that 10
If everything
years
goes
is
perfectly well.
if everything goes well, yeah. And that 10 years is full of paying for the salaries of the people who identify that drug, paying for the research costs and materials of testing the drug, first in dishes and then in animals and then in potentially in humans, right? Then you have to pay for clinical trials. So a phase 1 clinical trial, for example, right, where you're treating on the order of 30 patients, where you're just trying to see if how long the drug stays in the body and if there are any adverse effects. Like a phase 1 trial is just to understand how the drug works in a body. It's like $100 million, right? So then you have to imagine to go to phase 2, where you're treating, hundreds of people, and then phase 3, where you're treating tens of thousands of people. The cost skyrockets, right? And so it's very, very expensive to actually get a drug to market, because the levels of, or the expectations set forward by our government regulatory processes and global government regulatory processes is very high, because we want drugs to work, and the drugs need to do something better than what currently exists. It's not enough, financially speaking, or from the FDA in the US perspective anyway, to create a drug that just does as well as something that already exists, because otherwise there's no reason to introduce a new drug if it just only does as well.
Which limits competition.
It limits competition in the sense of companies are deprioritized to make something that does as well. But that doesn't mean, most new ideas, I would say, in like the biotech space, right, are trying to do something better in a way. I,
I
I,
see.
are you getting at this question of like sort of price gouging? Or
I'm not trying to set anyone up or anything.
no, no, I'm
I'm
just
just
curious.
trying to
Yeah,
ask questions.
I,
I'm
I
trying
want
to
to understand where
anticipate
you're.
what the audience might be asking. So let me ask you this. And sometimes, sure, I know the answers before I ask them, but
Hmm.
I know that that's not true for everyone. So another thing that I probably know the answer to, but I'd like you to talk about, is that when we think about drugs, there are types of drugs like Tylenol, which is acetaminophen, which is, let's say, a couple hundred grams per mole is the molecular weight of that small molecule drug. And so these are classified as small molecule drugs. And then in between, you have probably peptide therapeutics, which
Hmm.
can be on the magnitude of 1,500 to 3,000 grams per mole. And then you mentioned that Sanofi's claim to fame is this biologic, which is a monoclonal antibody, which can be on the order of hundreds of thousands of Daltons or grams per mole for the equivalent unit metric system. Can you talk to me about the price differences in the preparation and the distribution of a small molecule drug like Tylenol versus a monoclonal antibody or biologic drug?
Small molecule drugs like Tylenol, historically, or like penicillin for, like these kinds of things, like historically can be extracted like a very long time ago, right? These sorts of molecules could be extracted either from some plant or fungus or bacteria. And those are easy to grow, easy to harvest, and you're just removing something that already exists from a living organism. And synthetic chemists now, right, on the industrial scale have figured out ways to do this in very large batches from very basic starting materials. Biologics, monoclonal antibodies, are much more complex, as you allude to, and are much more challenging to make. And the engineering and process of building monoclonal antibodies is non-trivial, and it also isn't something you can synthesize from the ground up. It's something that you have to extract, and something that isn't naturally created by a cell or an organism. Right? You have to teach a cell to create this monoclonal antibody, and then harvest that monoclonal antibody and purify it. And so the production cost, as you alluded to, is actually quite substantially higher. Its storage also cost is substantially higher, because unlike acetaminophen or peptide therapy, which sometimes can just be lyophilized, I create it into like a powder, right? Monoclonal antibodies are not very stable like that. And so they have to be maintained, often frozen or refrigerated for long periods of time. And so there's a high cost of storage associated with these drugs as well. The big upshot, right, to using a monoclonal antibody is their specificity. And so because it's an antibody, it targets something very specific. And so off-target effects are much lower. And so in a way, it's a different class of drug. Um, Tylenol is a fun example because it has a very specific receptor, but there are lots of other drugs out there that have lots of off-target effects because they hit multiple different parts of the biological process.
That's exactly right. And I think a lot of people don't understand that. So this small molecule drug, those off target side effects are a result of basically this small molecule fitting into any sort of binding pocket inside of the body. Whereas this monoclonal antibody, so I'll give a famous example, is trastuzumab, which is a breast cancer treatment, specifically for breast cancer that overexpresses the HER2 gene.
Mm-hmm.
So this monoclonal antibody drug conjugate is will specifically go and target those cells that have that overexpression of the HER2 gene, and ultimately it will release the drug specifically to those cancer cells and only kill those, as opposed to the way a lot of chemotherapeutics work, which is basically just kill as many cells as possible and hope that we kill enough of the cancer cells that we can stop the treatment before you die.
Yeah. I think what's so interesting is that, as you point out, right, like this specific cancer drug specifically targets HER2 for this tumor type. And what you sort of are alluding to is like how complex these biologies are, right? And as much as like, there are simple drugs like Tylenol or um, Zyrtec, right, that have these conserved biological mechanisms, there are many more complexities and specificities to most parts of biology. And so we're sort of entering this new era over the last 20 years of um, a better understanding of biology, which leads to more focused treatments that can help specific people. So more limited in scope, but just as costly, unfortunately, to sort of build those drugs.
I think they sometimes refer to these as designer drugs, where they might even one day be able to make a drug specifically for you, even if you have the exact same disease as someone else. You know, occasionally, depending on your genetic makeup, we may be able to one day tailor drugs specifically to a person. Obviously, that's going to come with costs that hopefully will continue to decrease over time. But yeah, I think that that's a fascinating area of research. So, in addition to this work that you've done, moving from undergrad to graduate school to your postdoc work and now working in industry, you've also been working on side quests. And one of the ones that I know means the world to you is the work that you've stayed deeply involved with as it relates to what's known as URJ 6 Points SciTech Academy. And you've been doing this for years, not just as an alum, which you are, but also as a leader. And this is a Jewish summer camp that, from my understanding, focuses on science and technology. What did Jewish summer camp give you that elite academic environments didn't?
Yeah, Sci SciTech serves both boys and girls, but the the idea of URJ 6 Points SciTech is to provide a space for kids who um, don't succeed at traditional summer camps. And so I don't know if you attended a summer camp as a kid or not. It's more common, I think, in the Northeast and on the West Coast to attend summer
Sure.
camps. But the idea, right, is to find a community over the summer, give parents a break from their children, but also a community over the summer where kids can connect and experience things in a communal setting that is not an educational setting. And so in the Northeast, there are traditional summer camps where you go up into the mountains. You are in cabins with a whole bunch of other kids. You go on hikes. You play sports. But as you can imagine, when I was a kid, that was not what I really wanted to do. Right, I was not excited about playing basketball. I had asthma. I was a little overweight. And so hiking for 3 days and, in the Rocky Mountains in Colorado was like not fun for me. Um, and so with SciTech, URJ 6 Point SciTech Academy, which I abbreviate to SciTech, has done is it's created a space for kids to build those community connections, right? Like really forge connections with their peers around interests like science and technology, but where passion and creativity is completely free, right? Where you're not restricted to a curricula or sets of curricula that are decided to you, right? And the sky's the limit for what your interests and ideas can be. And you find like-minded kids who have the same feeling, right? And may not feel that they have those communities at home.
So, what is it that y'all do specifically?
At SciTech, we're a 2 week summer camp with multiple sessions, so multiple 2 week sessions. And what we do is we provide workshop opportunities for kids to build robots, design games, learn about biology. Uh, you know, uh, we've had herbology, we've had forensic science, we have programming and coding, and we also have like a digital arts and digital film. And the idea is that kids come and they take these workshops where they're doing this and working on a sort of cumulative project over 2 weeks, whether that be build a robot like a Rube Goldberg machine, like our 10th graders do, to build a big robot that does something very inane with 100 steps, or investigate a crime scene, a made up crime scene, and sort of create clues and trails. And then we also create a community where we value sort of like being ethical people. And so
What does that mean?
so the Jewish ethical values that we sort of like highlight at our camp are things like connection, respect, curiosity, discovery, um, and patience, right? And these values are not necessarily inherently Jewish versus another religion. I think they're inherently ethical or moral, and many religions engage with these, right? But what we really try to do is show that by, embodying these sorts of values, you can be a responsible scientist, engineer, and a responsible person, right? In the world, contributing to the world in a good way. And you can view all of this through a Jewish lens, and a science lens, right? These two things work together.
So you talk about, you know, doing ethical science and using Judaism as a guiding light in that process. What does that actually look like in practice when you're working with a 10-year-old?
Yeah, I think the the cool thing, right, is that 10-year-olds and kids in general, right, especially pre-high school, are really excited about the thing that they're excited about, whether it's building Lego or creating a video game program. And the way in which we sort of weave in the values is by framing their projects and their programs around these things, right? So when you're creating a video game, for example, often there has to be a sense of discovery, right? If you think about games that you play, whether it's that original Super Mario game, right? When you first hit that first question block and that mushroom pops out, right? That's a big sense of discovery. And when you finally touch it, right, and you become big, right? And
Sure.
so that those values we weave into the programming. And I think that's what's so special about it, is that we at the very base level help these kids think about their passions with these things that then they can take forward into high school and then in their careers, right? And we now have the camp's been around for 12 years. We now have alumni that are working in the biotech sector, in the tech sector, and contributing to these jobs with this outlook that they've gained from camp about doing things responsibly, doing them with respect, and openly, and trying to be good community members.
Has your experience mentoring young people through camp changed how you think about training scientists and building teams as an adult?
Definitely. I think that, um, campers are unfiltered, which I love. And what I think what I realize is that adults also want to be unfiltered, but you're sort of trained not to be. And the mentorship aspect of science is something that I very much love. And so I love that I get to do it with kids, and I love that I've gotten to do it with adults, right, or graduate students. I found that it's just really about giving people space to think out loud, um, and space to fail. And those I think are two really important facets of science, right? Which is experimentation, right? Making those observations and being okay that their hypotheses are wrong, and that just means they have to come up with a new hypothesis. And I love to try and help people work through these pieces, right? Because that's ultimately tussling with observation and making conclusions. That's the essence, right? That's the essence of what we do.
You seem really passionate about this.
I love it, and I love the kids, and I love that there's a space out there for kids like me, who didn't necessarily fit in other places, and so for the opportunity to come build robots, or build camp in Minecraft, or launch rockets, like to do that and then also experience this community building aspect of things, I think is really special.
Yeah. So, Sam, if you don't mind, I'd like to change gears pretty substantially. And there's something that I want to ask you. You are a Columbia alum and you're a Jewish scientist. When I think about Jewish and Columbia, I immediately think of recent events that have happened in the country, but specifically on Columbia's campus. How have you processed watching universities become political flashpoints in the Trump era and maybe even beyond?
I feel deeply connected to Columbia and my Jewish experience at Columbia. I was very involved in Hillel there, which is a Jewish organization for college students. Um, and I think what's become very challenging to parse as someone very involved, in that life while on campus, is uh, where messaging comes from, right? And what's real and what's not real. I think even in the pre-AI era, we look back, pre-mass AI era, like we look back at like 2022, 2023, and sort of the things that were happening on college campuses, right? It's hard to understand, right? There are claims of anti-Semitism from students, there are claims of anti-Semitism from faculty, and there are, claims of, uh, uh, racism against, Arabs and other other Middle Eastern ethnicities. And so it's challenging because, as we've sort of learned, is like these mass demonstrations at Columbia and other campuses are a lot of people that are not students, that don't represent the universities. And I think that's a key point that's lost a lot, is how complex these problems are, and the way in which they're actually unfolding is often much broader context than the university. And it's really hard to know the truth because it's just being reported and then re-reported by other outlets, right?
Is that what you think people outside of academia most often misunderstand about what's happening on college campuses?
I think because there's so much information that we process in the day to day, everything needs now to be compressed into one or two sentences, right? When I look at news, I'll be like completely honest. When I look at news, I get a push notification on my phone from the news organizations that I subscribe to. I read the header, and unless I'm really invested in that article, I'm not going to read the article. Right? There's not enough time in the day anymore to do that. And so, because everything's compressed, people take away the information that's given to them, that it has a slant, has a bias from the way in which it's presented, and takes away all of the nuance. And I think Columbia has been really unfortunate in becoming politicized in terms of, yes, like one versus the other. And I think the discourse that is not happening on campuses that needs to be sort of like prioritized is the gray area discourse, right? It can't be yes or it doesn't have to be this or this, right? There can be relationships in the middle or communication in the middle that are important to capture.
In this era, there's been a lot of using funding as kind of the stick or the carrot as it relates to what sort of policies to enact on different institutions. And and I think to the general public, this depend actually probably doesn't actually depend on which side of the fence you are on politically. You don't maybe see this as having a huge impact, but what people don't understand is that most of the science that ends up coming out and being on the shelf when you need to take a drug or a medicine at some point has worked its way through an academic institution where they either looked at the system at which this drug is behaving in a certain pathway, or maybe it was discovered and developed initially at a university in a research lab, which is funded by public dollars. Can you talk to me a little bit about why we should care about what's happening with funding science in the United States right now?
I think my favorite example of this is how MRI came about, right? Magnetic resonance imaging. And MRI is based on a spectroscopy technique called nuclear magnetic resonance, which Dr. Rojas, you're very familiar with, which is a way to sort of look at the shape and state of a molecule, right? And this, in and of itself, was actually a physics discovery. It was in a physics lab where they came up with this idea of trying to measure a molecule using magnets, right? That then translated into a chemistry problem or a technique that then translated into a biological problem or technique that's used very broadly in the medical field, right? And so the initial research being done had no connection to the medical field initially to create this technology that then had very substantial long-lasting impacts on medicine and our capacity to diagnose diseases and disorders, right? And so I think basic research, and often that's what's being at many universities, as was covered by government funded grants, could be on something that has no impact, but it could also no immediate and understandable impact on medicine, but could also be funding something that has substantial, long-lasting impact. And I can speak from like the pharma standpoint of things, is like, we have the ability to generate data, but we can't take risks like uh, academia can, right? Our bottom line has to be that we have to pay the bills. We have to keep the lights on so that we can continue to make drugs. But we can't ask open questions and take risks like academics can. And that's the big value in funding academic science, is that they can feel free to explore things that we can't. And, there's an ongoing debate about which is better or more efficient. And I don't necessarily want to dip my toes into that, but I think if you're interested in that, I would say, you know, you should read people should read about this conversation about what's more efficient. Is it the sort of freedom to explore, or really um, directed testing of kind of like new molecules or new tools?
To give another example of one that's in the popular narrative right now, and that's GLP-1s, right? And
Yeah.
these are the weight loss drugs that people are taking, which came from a gut hormone discovered while scientists were trying to explain a weird observation that oral glucose lowers blood sugar better than IV glucose. And that mystery led to the idea of what are known as incretins and the discovery of the GLP-1 eventually. And ultimately, this all goes back to finding it at, like, initially like in Gila monster or something that scientists working on not weight loss drugs found, right? And had no
Yeah.
intention of developing some sort of drug molecule. And I think that that's a big thing that people don't understand is that, sometimes the greatest discoveries that actually matter to the most people. I mean, there are millions of people using GLP-1s right now, many of them for medically necessary reasons, and we owe that all to some like random chance discovery because we let a scientist roam wildly in the world and just be open to finding new things. So to wrap up, if you could talk to the version of yourself right before starting your PhD, what's one belief you'd want to unlearn?
That's a great question. Um, I think the belief I'd want to unlearn is that I have to know what my next step is going to be.
So
I
be
think
okay walking in the dark.
be okay walking in the dark, be okay, you know, taking turns, even U turns sometimes, right? And that um I think something that you don't learn, or at least I didn't learn until late enough in my career, is to follow the science, right? And that really translates to follow your instinct, right? Look for those open doors. Don't be so boxed in by the thing you think is going to be best for you, or the thing that you think is going to, like, like I said, your hypotheses can be wrong, and that's okay. That's good. Um, and so I would want to unlearn that need to feel like I need to know where I'm going, and I need to know what's going to happen, and I need to know what my life's going to be like. Because at the end of the day, like, there's so many opportunities that are going to arise in every person's life, and being open to those, I think, is the most important thing.
For students who love science but also care deeply about community, identity, or impact beyond papers, what's your advice for not feeling like they have to choose one or the other?
I think that within science, there are so many great ways to um, be a mentor, get involved in um, the journalism aspects, or news coverage, or policy. These are all things you can do within your training, which is great, and these opportunities exist. I also think that, for me, it's been personally fulfilling to find something to do outside of work that brings you joy, whether that's bartending at a student bar on campus like I did when I was at MIT, or volunteering your time to work with kids in a community who don't have lab science classes or things like that. These are all opportunities, and it's important to keep yourself sane, right? Like, you can only spend so much time thinking about school and work, and you have to bring yourself some fulfillment as well in your personal life.
Thanks, Sam.
Yeah, thanks so much, Anthony.